Our next speaker is Dr. Geoff Foster from Scotland's Royal College. Geoff is stepping in for Dr. Andrew Webb at the last minute because unfortunately Andrew is unwell today. So thank you very much, Geoff. Geoff's a microbiologist and has kindly volunteered to go through Andrew's slides for us. Unfortunately, I know Geoff can't stay for the Q&A session, so if you have, please do put your questions directly for Geoff into the chat as he talks through his presentation, and we'll pick up any questions directly after his talk rather than waiting for the Q&A slot. So if Geoff's on the call, if I could ask you to share your slides, Geoff.
This is work that was funded by the VMD between ourselves and Andrew Webb at the Scottish Microbiology Reference Laboratories in order to look at Staph pseudintermedius that we'd collected over a 14-year period. Staph pseudintermedius is one of the members of the former Staph intermedius group and the broader intermedius hyacus group. These organisms all at the top there, all the way down to coagulans, are all coagulase positive species. There are 850 isolates in the collection that we used for this. Oh, sorry, didn't realise animation was going to be on here. And that covers the range of species that we have had Staph pseudintermedius from. So we had 5% of the isolates were human. This was 406 isolates in total, 366 dog, 31 human, 8 cat and 1 otter were all methicillin resistant. These were the antibiotics that were tested against. Many of these are for human purposes rather than what would be used in animals. We found that of all the Staph pseudintermedius, 96% were resistant to at least one antibiotic that was tested. There was no resistance to rifampicin, vancomycin, or teicoplanin, which are very significant organisms in human work, but not used in animals. There were 53 different resistance phenotypes, and 40% of these were seen only once. And 43 phenotypes were actually MDR. We had significantly higher rates of resistance to chloramphenicol, ciprofloxacin and enrofloxacin, clindamycin, erythromycin, gentamicin, tetracycline and trimethoprim in MRSPs than we did have in sensitive pseudintermedius. The fusidic acid rate was higher in this methicillin sensitive species, which was a bit unexpected. Looking at the graph here, you can see everything in red is an MRSP, and as you would expect, the columns are much, much taller there, and complete resistance for oxacillin and penicillin, of course. MSSPs varied a bit, but some of them were still quite high. You can see a very high level of penicillin resistance there, even to MSSPs. Overall rates of resistance were similar to other European countries. 16% were MDR. We had 1 XDR isolate. The most common pattern was penicillin, fusidic acid, and trimethoprim. In this graph here, you can see the variation over the years 2015 to 20, and mostly it is creeping up slowly, with the exception of trimethoprim and the red one, which is fusidic acid. The reasons are unclear why we have a temporal variation in the resistance. It may be one of the things towards the end of the study is, of course, there were many more dogs around following COVID. And could it be related to the use in companion animals or a changing population structure? Pet ownership, as you can see, has gone up in cats and dogs following the COVID outbreak, or even starting to rise perhaps a bit before that. Press the wrong one there. Antimicrobial resistance genes. There were also resistance to quaternary ammonium compounds found, particularly almost as strong in the MSSPs as they were in the resistant strains. Okay, he's got me a little bit here. I think he's talking mostly about the quaternary ammonium compounds there. So as we said earlier, all oxacillin resistant strains were MEC-A positive. There were no MEC-Cs. Apologies, I've got a grandchild crawling over me here at the moment, if you hear something else. 92% of the carried it. 92% of this must be the oxacillin resistant ones, carried the SCC MEC element. 7 of these were non-typable, and there were 3 particular SCC MEC types, 2, 3, 4, and 5. Genotyping, we had 85 different STs identified. 50 of these were novel. And the MRSP clonal population, I'm not quite sure what he's wanting to add there. Maybe this is it. Genotyping for MLST, ST71 is by far the most common in Europe. ST68 found most common in North America, and ST45 and 112 in Australia and Asia. So interesting to see how ours compare with these places. On the MRSP genotyping, we had 19 isolates were singletons, and ST71 was indeed the predominant ST, which we would have expected, as that's the most common one in Europe, followed by ST68, 726, and 551. MSSP were more heterogeneous. 93% of them were singletons, and only 3 strains were actually occurred more than once. This is going to be a bit tricky here. ST, the colours down the left, I think, are down here. The colours, that pertains to the host animal species, I think, and the ST551s were all from cats, the yellow ones there. Not a lot more I can say about that because it's very small on my screen as well. Yeah, so you can see that the 4 different main methicillin resistant ones there. 2 of the clones were new to Scotland. ST726 appeared in 2016, and ST551 first appeared in 2020. While second emerging clone ST551 was not detected until 2020, and we had 4 strains, one of which was human. ST551 is an emerging clone in central Scotland. And what we think has happened is that it has came with imported dogs from Slovakia, where it had been reported several years earlier. And that probably reflects the fact it didn't appear until 2020 as a period when people started getting a lot of dogs in from Eastern Europe. And that is reflected in other things such as raw pet food issues. Oh, and that seems to be it. Thank you. Sorry about that. It's all been a bit hurried this morning to put this together, but I'm happy to take any questions.
Thank you so much, and no, huge thanks for stepping in at the last minute. Brilliant job. I will hand over to my colleague Tamsin Dewey, who's the head of antimicrobial resistance surveillance and evidence within the AMR team here, and she's going to be chairing the Q&A sessions. But I'll hand over to her to field any questions that are coming in for you specifically, Geoff.
Thanks, Katrina, and well done, Geoff. I know how impossible it is to take on board somebody else's presentation, especially when it's full of technical details. I just wanted to start with a little bit of contextualization, perhaps for some people in the audience, and then I'll come to a question for you, Geoff. So my understanding is that Staph pseudintermedius, which is the bacteria you're studying here, this is the major staph in dogs, right? Whereas people in the audience might be more familiar with Staph aureus, which is from the same family, you can say, but is more commonly present in people. I just wondered, could you comment on, or do you know what the likelihood is of resistance genes being able to transfer between Staph pseudintermedius and Staph aureus?
I don't know much about individual genes that have been transferred. However, what I would say about Staph pseudintermedius is that it's now being found in humans more frequently. In the past, it wasn't being found at all because of the way that they look for Staph aureus in hospitals, which was basically a latex test. And most Staph pseudintermedius would have been getting reported as coagulase negative Staphylococcus, which they obviously are not. Because of MALDI-TOF, at least the Bruker one identifies Staph pseudintermedius. The biomaerial one, I gather, does not. And I've been pushing for 20 years on this, that Staph pseudintermedius must have been going to humans because it's the reverse of what's happening with dogs getting Staph aureus from humans. And so you would think there's certainly an opportunity, at least for a swapping of genes between them, but I'm not aware of any actual published information on that.
Okay, brilliant. Thank you. I think that's a helpful generalizable point that AMR transmission happens in both directions, but from animals to people and from people to animals back again. And the more we know about it, the better place we're going to be to make targeted interventions. So thank you very much. Sorry, go ahead.
Just one thing I would add there. If you happen to have a diabetic foot ulcer, don't let the dog lick it. That's where a lot of these things are coming up.
Thank you. That is an excellent public health message. Thank you, Geoff. I'm going to hand back to Katrina.
Thank you, and thank you again, Geoff. Sincere thanks from everyone at the BMB. Great job stepping in there.